Okay, the blood has several purposes, and we're going to look at four of them today. We're going to look at four major things the blood has to do, and and of course the blood. When we're talking about blood, we're talking about the circulatory system, and that's because that's the system that actually moves the blood around the body. The blood has four major purposes, and there are more, but the four big ones that we're going to talk about today. And the first was bringing in a couple things. The two things it definitely needs to bring to the tissues is oxygen gas, and the second thing it has to bring to the tissue is it has to bring in food. Okay. The second thing, the other thing the blood has to do is take things out away from the cells of your body that make you who you are, your brain, your toe cells, your muscle cells. It has to take out first carbon dioxide, and then it has to take out waste. Okay, it has to somehow treat the waste, get it out of your body. When we look at the circulatory system, you'll see, let's start at the heart. Let's start here. Remember, it's a circulatory system, so it's a big cycle. All the blood is inside. Here's a cross section of the heart. So the heart's been cut in half. And you'll see that, again, I want to emphasize this is already in your notes, but all the vessels leaving the heart are called arteries. All the vessels coming to the heart are called veins. So here from the right ventricle, that's this name for this big chamber here. The two big chambers are ventricles. The two small chambers are atria or atrium. From the right ventricle, you'll see that the blood goes and splits into two arteries. This is the only artery that has deoxygenated blood, or blood that does not have oxygen. And it's going to go to each of the two lungs, where oxygen is going to come into the blood and carbon dioxide is going to go out. How is that going to happen? It's going to happen at the capillaries, at the tiniest little blood vessels down here, and we'll take a look at that in a minute. And you've already taken a look at that a little bit on your online activity. It has to happen through diffusion, because remember, carbon dioxide and oxygen diffuse through the membrane with very little trouble. And so carbon dioxide leaves because the carbon dioxide is higher concentration in the blood and less concentration in the lung. So carbon dioxide goes out. Oxygen has low concentration in the blood and high concentration in the lung, so it goes into the blood. So you have simple diffusion happening at the lung. Then, as the blood is pumped, as this muscle squeezes, the blood gets moved in here and then goes through the capillaries back out and back into the left atrium. That's the small chamber here. And then from the left atrium into the left ventricle. When the left ventricle squeezes, that's when the blood then shoots out through the aorta, and with full of oxygen, the blood had already stopped over here at the kidneys and the liver. At the kidneys, it took care of the waste. Okay, and the waste got filtered out, and it was sent to the bladder where you urinated out. And then the CO2, of course, we just got rid of it here at the lungs. The food came in already here at the liver, or here at the intestines. It would be around here, around the portal veins. All the food, all the blood that was going in around the intestines, the intestinal nutrients diffused or were brought in through different proteins into the blood, and that blood with all the nutrients and all the food then went into the liver where it was processed and then put back into the blood again. So we got the food in there, check. We got the waste out, check. Carbon dioxide's out, check. Oxygen's in, check. So now we got to get this blood that's full of nutrients, has carbon dioxide taken out of it, has the waste filtered out of it. We got to get this just nutrient-filled, oxygen-filled blood, waste-depleted blood going out into the rest of the body, and that happens from the left ventricle into the aorta. See here the two carotids going up into the two brachiocephalic arteries going up into the different places in the head, which would be over here, and then the aorta going out and feeding the rest of the body, and branches, and keeps branches like a big tree, feeding every cell of your body with this high oxygen, low CO2, high food, low waste blood. Of course, at some point in the branches, it goes right back into the veins, and it heads back over to the kidneys where it takes out the waste and goes to the liver. It goes to the intestines where it picks up the food. It goes to the liver where the food is processed, back in the blood, back to the lungs, etc. So this is a big cycle. So just a real close look at it. There's your heart. Now please notice these tiny little veins and arteries. The arteries, at some point, there's these tiny little things called capillaries there that connect the arteries to the veins, and we'll see that in just a minute. But this is the heart, you see, and here are the lungs, here's the aorta, here's the vena cava, okay? So the blood's coming back to the heart, all right, through these veins, and it's going away from the heart here through these arteries. Here's the left common carotid, and here's the right common carotid, and here's the some more larger branches here, and that's how the blood circulates. Now, as I said earlier, there's these tiny little vessels. There's a connection between the artery here, the red part, and the veins, which are the blue part. There's a place in which the two connect. They get smaller and smaller and smaller as they move down the artery. The artery gets smaller and smaller and smaller until it's just 1 or 2 cells wide, and then it starts to feed back into the veins. So the blood comes in with all the nutrients, all that stuff's diffusing, the waste goes into the blood, the oxygen and the food goes out of the blood, and so you end up with this blood that's in the veins that's depleted of oxygen, is full of carbon dioxide, or not carbon dioxide, excuse me, full of waste, the blood is, and the carbon dioxide is in the blood as well, and that goes back to the processing units again. Now here's a closer look at a drawing, but what's cool about this drawing is you see that the veins and arteries are built of cells. They're not like some plastic tube. Each of these tubes are built of cells all the way around. And you'll notice that it just branches. You see the branches keep happening all the way down, and the diffusion's happening here. High concentration stuff's going to go into the blood. High concentration stuff's going to go into the tissues, and back and forth until dynamic equilibrium's made. The same thing's going to happen as the veins go back out. Now, so here is the artery. You'll notice that this is a cross-section of the artery. Again, it's made of cells. So you see there's a cell here. These are the cell nuclei. The artery is actually made of cells. It's lined by cells. And, of course, the tissue's here. So this tissue could be lung tissue. It could be liver tissue, kidney tissue, muscle tissue. Any organ you name it, everywhere where there's cells, you're going to have some artery and some vein associated with it, where you're going to get the stuff from the inside that's good going into those tissues, and the stuff that on the outside from the cells that the cells need to get rid of is going back into the blood. The blood is carried here inside this tube. It loops around after it reaches a capillary, and it comes back here in the vein. Notice the arteries are thicker. They have much more muscle to them. They actually keep their tenacity longer. They stay very toned, very rather rigid. They can hold a lot more pressure, where your veins can hold a little less pressure. They're a lot more flaccid because they have less muscle. Okay, so what's happening at the cellular level when we're looking at this barrier between the blood out here and the cells? Okay, so this membrane represents the barrier between the blood. Remember, there's the plasma or the serum or the plasma of the blood, and then there's blood cells as well. But here's a cell that represents the barrier on the other side, which over here on the other side there's another membrane, and down here in this other membrane there are other cells. And so there's going to be diffusion and movement of substances from the blood across the membranes, and all of them are going to just keep going across, and they're going to be spread because you're going to get this dynamic equilibrium happening. But along this membrane, you'll see that there's a high concentration of these yellow triangles. Here you have a transport membrane that's going to membrane protein that's going to allow things to travel, high concentration, in this case triangles, to travel from the inside to the outside. Remember this, every time you see any kind of drawing, things are going to move from high to low, assuming they can. If they can't, then water will travel through a very similar protein to this one that's going to allow water to come in and fill up and regulate that concentration again. You notice the blue parts are all hydrophilic. They all represent the kind of things that are dissolved in water, where the tan parts here are all nonpolar, all right? So they don't dissolve in water. Here you have a cholesterol molecule, and over here you have this one. This protein does not cross the membrane, has a big carbohydrate chain on it. Notice it's long, it probably acts as some kind of identifying protein. So there's different functions of proteins. If we take a look at this, concentration gradient here, you have a high concentration. Here's low, so the direction of the concentration gradient is this way. Is the direction that the dot is traveling. So you go from high to low. That is your concentration gradient. You have transporters. They can transport using either ATP or passive diffusion. So they could either be facilitated diffusion, which does not require ATP, is passive diffusion, a passive transport. And then you have ATP-driven transport, so it's active transport. And active transport is when you're going against the concentration gradient. So again, here you have your transporters, which are moving things across the membrane. If you think about function and structure, the structure of the transporters transporters must cross the membrane. They also must have an opening. They have to allow things to go in and out. Anchors are just exactly what they seem. They're things that are embedded in the protein. Usually they have parts that are hanging out, and notice how this is kind of all tied together. This anchor could hold two cells together. It could hold a very large protein or sugar chain to the cell. It could do a lot of things that cells need to do where they require something to stabilize a certain area. Receptors, well, here you have a blue part that's waiting for these red molecules. The red molecule could be a hormone. The red molecule could be a chemical, like acetylcholine, a neurotransmitter. But whatever it is, this red molecule is going to be accepted by this blue protein that's across the membrane. When that red molecule's there, it signals the cell, and we're not going to go into how that signal happens, but it's a chemical signal. It changes something down here. Remember, change or difference is information. Difference is actually the essence of information. So it changes the inside. By making a difference in the inside, it sends a kind of a signal to the cell. The cell then does something. It opens up one of the transporters, or it starts up an enzyme, or it starts making a protein. Whatever it does, this signal starts when that molecule connects to the receptor. And we'll talk, a really great example of that is insulin and diabetes, and we'll be talking about that soon. Here's an enzyme. An enzyme can also be embedded in the membrane. So here's this embedded enzyme, and here's an enzyme where every time X comes along, it makes a Y, and so you're going from X to Y. Remember, enzymes themselves are not changed, so this enzyme could sit in the membrane making Y all day long. Well, why would it be embedded in the membrane? Maybe Y is needed in the membrane, or maybe Y maybe this tail acts as part of a signal machine, and maybe the one of the other proteins signals the enzyme to start working by changing its shape or changing its conformation, making it active. And at other times, the switch can be turned off. So these receptors can actually act like switches. They turn enzymes off or on, or they can act like switches for the DNA, the nucleus, to start making proteins or turning them off. So they're pretty interesting, fairly interesting little structures. Remember, they're proteins that are embedded in the membrane, and so you can have these proteins that are embedded in the membrane can have many different functions. These are just four of the simpler ones, and they're pretty good general categories. All right, that's it for this this little video. I hope this helped.